Hello dear students, today here again we are with a new video of biology from chapter number 4, molecular basis of inheritance. Topic is Hershey's and Chase experiment. In my previous topic, you have seen about the Avery, McCarthy and McLeod. They have tried to prove that the DNA is a genetic material for transforming principle. But Hershey and Chase was having a doubt. Therefore, they started proving the experiment again with the different kind of the varieties of the strains. When Hershey and Chase started trying the experiment, what they did, they worked with the viruses, which was not tried by Avery. So they started trying the same experiment to prove the DNA as a genetic material for a transforming principle. They started using viruses which infects the bacteria, that is bacteriophage. This is a new word for you. Bacteriophage is the strain of bacteria where they are composed of DNA and proteins. Bacteriophages. So they used the two different kind of radioactive materials. One was radioactive phosphorus and another is radioactive sulfur. For radioactive phosphorus, they have used 32 phosphorus strain, and for radioactive sulfur, they have used 35S sulfur, that is radioactive sulfur. You know the radioactive substance emits the radiations from it. So when these two different radioactive materials were used in the medium for culturing the strains of R and S type, so what they found that they used this in medium for some viruses. The viruses which were being used for infecting the bacteriophages. So when they used radioactive phosphorus and radioactive sulfur, the results were shocking. And they found that the viruses grown in 32 phosphorus shown radioactive DNA. Means when the virus's culture was being inculcated in the medium of phosphorus 32, the radioactive DNA was isolated, and they found that this DNA was labeled with radioactive material. But when the same experiment was conducted with the sulfur 35S strain, that is the radioactive sulfur 35, they contain radioactive proteins, but there was no radioactive DNA. Means only the outer covering, that is the coating, was being radioactive material, but DNA was not being coated with the DNA, that is radioactive material. So they found that the viruses which were coated with phosphorus were responsible for transferring the genetic material. So for proving this, what they did, the bacteria E. coli was used first time. E. coli is the bacteria which is known as Escherichia coli, which is a very common bacteria which is easily grown in an artificial medium, we can say laboratory. So E. coli was used to get infected by the radioactive phages. So the radioactive phages which was used, which was already been cultured in radioactive phosphorus and radioactive sulfur. So viral coats were removed by using which method? Centrifugation method. Centrifugation is the method where the strain or the sample is being rotated in the rotation per minute, that is RPM, with a high speed, so that it gets separated with the supernatant and the pellets at the bottom. So what happens? Only the radioactive phosphorus was found inside the bacterial cell, indicating that the DNA is a genetic material. The confusion was that whether DNA transfers or whether the protein is transferring. Because in starting I have said the bacteriophage generally is composed of two materials, DNA and proteins. So they were in confusion whether protein is responsible for transformation principle or whether DNA is responsible for transformation principle. So for this they concluded one experiment. When the phage is being infected and the infected phage will be treated with the 32 phosphorus, the DNA core was being added in the E. coli bacteria, and when 35S sulfur code, that is the protein code, was being infected with the E. coli bacteria, what they found? After blending and centrifugation in both the steps, they found that the phosphorus was detected in the cell where phosphorus 32 was used. And phosphorus is generally the material which is found in the DNA, that is DNA level material. And in sulfur coating, the phosphorus was not found, neither the sulfur was found. So they proved that the cell with the supernatant sulfur was there, but here the DNA was being labeled with the phosphorus was found in the cell. So they concluded that the DNA is the only material which is responsible for the transforming principle. In this way they proved the theory that the DNA is a genetic material. I hope you understood the lecture. Thank you for watching the video.